Hello viewers, my name is Mugaiga Senunamayo, and today I'm going to discuss hyperplasia, metaplasia, and dysplasia. To begin with, hyperplasia. What is hyperplasia? Basically, this is the increase in size of a tissue or an organ due to the increase in the number of cells. Or somebody would simply say is the increase in the number of cells of an organ. Then it will only occur in labile and stable cells. The labile cells are the cells that are capable of continuously proliferating, while the stable cells can pause cell division, and there is another group of cells called the permanent cells that once they have matured, they can never undergo cell division again. For example, you have the neurons and the skeletal muscle cells and the cardiac muscles. Then the mechanism of hyperplasia is that you will have increased proliferation of the stem cells, and once you have this, you will have an increased output of new cells. Then number two, in mature cells, you will have growth-induced proliferation. Then hyperplasia has two types. The first one is physiological, the other one is pathological. Physiological one occurs as a response to a normal stressor. For example, the thickening of the endometrium during estrous cycle and enlargement of the breast during puberty, pregnancy, and lactation. But it also has another form called compensatory hyperplasia, which occurs after surgery. For example, part of your kidney or liver has been removed. Then the remaining cells in the organ will undergo proliferation and cover up that missing area which has been removed. Then for the pathological hyperplasia, it occurs as a response to an abnormal stimulus like hormonal excesses, also in neoplasms, then also in benign prostatic hyperplasia. Moving on to metaplasia. Metaplasia is the reversible replacement of one form of adult cells by another form that is not normally found in that tissue in response to normal or abnormal stimulus. Then these stimuli can be physiological or pathological. Hence, metaplasia has also two types, which may be physiological or pathological. Then metaplasia itself is benign, in other words, non-cancerous, but however, if left untreated, the cells undergoing metaplasia will become dysplastic, and which will eventually lead to cancers. Then there are also two types of metaplasia, which is epithelial and mesenchymal. First, let us talk about epithelial metaplasia, and this is the most common type of metaplasia, and whereby new epithelium is stronger but less well specialized to meet the functional needs of that particular tissue. Then depending on the new cell type, there are two types of epithelial metaplasia, and that is where the metaplasia gets its name. For example, we have squamous metaplasia, where various specialized epithelial cells are capable of changing into stratified squamous epithelium due to chronic irritation like mechanical trauma, chemicals, or infections. This is well seen in the respiratory tract of smokers, where the pseudostratified columna is replaced by stratified squamous epithelium. Then number two, we have the columnar metaplasia, where the new cell type is simple columnar. We see this in Barrett's esophagus, and normally in a disease called gastroesophageal reflux disease, where we have gastric acids getting back to the esophagus. So normally the squamous cells are not resistant to these gastric acids. So what the stem cells will do, they will start proliferating, but as they are proliferating, they'll change into columnar cells, which are more resistant to the gastric acids. Then we have the conversion of pseudostratified ciliated columnar epithelium in conditions of bronchitis and bronchiectasis to simple columnar epithelium. Now, the left slide is showing is an example of glandular metastasis. This is just a form of columnar metastasis, whereby we have this is the esophagus, whereby we have the stratified squamous epithelium being replaced by simple columnar or the glandular epithelium of the stomach, as you can see. Then the right picture is showing the normal gastroesophageal junction. As you can see here, we still have our stratum basal very clear, but here it's getting lost. And immediately below the stratum basal in the normal, we have lamina propria, but here we are having glandular cells. We are having like tubular glands in that area. Now we have mesenchymal metaplasia, which is a more rarer form of metaplasia to epithelial metaplasia, and in it we have osseous metaplasia, which is the replacement of non-osseous soft tissue by bone, and we can see this in ossification of tendons and cartilages in older turkeys and horses and draft animals. So this is just because of age. Then we also have ossification in the lung tissue, just as these pictures are showing. First, this is the lung parenchyma. Also here, this is the lung parenchyma, and same thing for here. Then this focal region here is a region of ossification, whereby we have unmineralized bone tissue forming in the lung. Then this membrane here is the pleural membrane, and still this is the lung parenchyma, and we have an unmineralized bone forming here, but it has a mineralized bone portion, as you can see that. Then this focal point here is completely mineralized bone in the lung parenchyma. Still under mesenchymal metaplasia, we have cartilaginous metaplasia, also known as chondrometaplasia, which is the replacement of tissue by cartilage. Then in appearance, we can see cells that are residing in lacunae, like these ones, and then they have nuclei that are dark, single, and small, and they don't show any mitotic activity. Then lastly, we have dysplasia, and dysplasia is simply abnormal development or growth of cells in tissues, and this leads to loss of uniformity in the cells and loss of structural orientation. Then, for example, in dwarfism, long bones fail to grow to their normal length because the growth plate is highly disorganized, and this occurs in tissues that are constantly renewed, like skin and mucous membrane, and It may occur during fetal or neonatal development or in adult tissues that are constantly being replaced. And genetics is the most common cause of this dysplasia. And dysplasia is characterized by disruption of structural orientation and polymorphism, which means that there is more than one distinct form of cells when you view it. Then we have the hypochromatic nucleus, which means it is darker under stains, and increase in nuclear cytoplasm ratio, which means that the nucleus is kind of larger than normal when you compare it to the cytoplasm. And we have a lot of mitotic figures, which shows that these cells are in constant cell division. And another thing about dysplasia is that it precedes cancers. Well, then this picture is showing moderate endocervical dysplasia, whereby normally we know that the cervix or the endocervix has a simple columnar epithelium which is ciliated. You can tell that this is the normal region around here because it has long cells with the nuclei located at the base. But if you look at this area here, you can't really tell the shape of the cells. You can't even tell whether the nucleus is spherical, because in columnar cells, the nucleus should be spherical. But these cells, the nucleus doesn't show like a spherical shape, and they are highly disorganized. You can't even tell their shape. So thank you for watching. That is all I had for you. Please subscribe to our Innovates channel and like and share.